हेलो एंड वेलकम बैक टू माय चैनल दैट इज़ द एनसीईआरटी ऑडियो बुक। सो आज का जो हमारा चैप्टर है वो ह्यूमन फिजियोलॉजी का दूसरा चैप्टर है दैट इज़ ब्रीदिंग एंड एक्सचेंज ऑफ गैसेस। और एनसीईआरटी के अकॉर्डिंग ये चैप्टर 17 है। सो हम लोग स्टार्ट करते हैं। As you have read earlier, oxygen O2 is utilized by the organisms to indirectly break down nutrient molecules like glucose and to derive energy for performing various activities. Carbon dioxide, that is CO2, which is harmful, is also released during the above catabolic reactions. It is therefore evident that O2 has to be continuously provided to the cells and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hand on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following sections of this chapter. And our first topic is 17.1, that is respiratory organ. Mechanism of breathing vary among different groups of animal depending mainly on their habitats and level of organization. Lower invertebrates like sponges, coelacanths, flatworms, etc. exchange O2 with CO2 by simple diffusion over their entire body surface. Earthworms use their moist cuticle and insects have a network of tubes, that is tracheal tubes, or to transport atmospheric air within the body. Special vascularized structures called gills are used by most of the aquatic arthropods and mollusks, whereas vascularized bags called lungs are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin. Also, mammals have a well developed respiratory system. Moving to the next topic, that is 17.1.1, that is human respiratory system. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which help in sound production and hence called the sound box. During swelling, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of the food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity which divides at the level of fifth thoracic vertebra into a right and left primary bronchi. Each bronchi undergo repeated division to form secondary and tertiary bronchi and bronchioles, ending up in a very thin terminal bronchioles. The trachea, primary and secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous ring. Each terminal bronchiole gives rise to a number of a very thin irregular walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles, and alveoli comprise the lungs that is given in figure 17.1. We have two lungs which are covered by a double layer pleura with a pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in close contact with thoracic. And in 17.1, diagrammatic view of human respiratory system, that is sectional view of left lung, is given. Thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts form the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to alveoli. Clear it forms foreign particles, humidifies, and also bring the air to the body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lung is situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by ventral column, ventrally by sternum, laterally by the ribs, or on the lower side by the dome-shaped diaphragm. The anatomical setup of the lungs and thorax is such that any change in the volume of the thoracic cavity will be reflected in the lungs, that is pulmonary cavity. Such an arrangement is essential for breathing as we cannot directly alter the pulmonary volume. Respiration involves the following steps. First step is breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2-rich alveolar air is released out. Second step is diffusion of O2 CO2 across alveolar membrane. Third step is transport of gases by the blood. Fourth step is diffusion of O2 and CO2 between the blood and tissue. And the last one, fifth step is utilization of O2 by the cells for catabolic reactions and resultant release of CO2, that is cellular respiration, as are dealt in the chapter 14. And the next topic is 17.2, that is mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric is drawn in, and expiration, by which the alveolar sac is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, that is intrapulmonary pressure, is less than the atmospheric pressure, that is there is a negative pressure in the lungs with respect to the atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized setup of the muscles, external and internal intercostals between the ribs, help in generation of the such gradient. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the anterior posterior axis. The contraction of external intercostal muscles lifts up to the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to the less than the atmospheric pressure, which forces the air from outside to move into the lungs. That is inspiration. That is given in figure 2.17.2a. Relaxation of the diaphragm and the intercostal muscles returns the diaphragm and the sternum to their normal position and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, that is expiration, that is given in 17.2b. We have ability to increase the strength of the inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 minutes times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. And the next one is 17.2.1, that is respiratory volumes and capacities. And in figure 17.2, mechanism of breathing showing inspiration and expiration. First one is tidal volume, that is TV. Volume of air inspired or expired during a normal respiration. It is approx 500 ml. That is, a healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. Next one is inspiratory reserve volume, that is IRV. Additional volume of air a person can inspire by forcible inspiration. This averages 2500 ml to 3000 ml. Next one is expiratory reserve volume, that is ARV. Additional volume of air a person can expire by a forcible expiration. This average is 1000 ml to 1100 ml. Next one is residual volume, that is RV. Volume of air remaining in the lungs even after a forcible expiration. This averages 1100 ml to 1200 ml. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity, that is IC. Total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume, that is TV plus IRV. Expiratory capacity, that is EC. Total volume of air a person can expire after a normal inspiration. It includes tidal volume and expiratory reserve volume, that is TV plus ERV. Next one is functional residual capacity, that is FRC. Volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, that is VC. This, the maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV, and IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity. Total volume of air accommodated in the lung at the End of the force inspiration. This includes RV, ERV, TV, and IRV, or the vital capacity plus residual volume. So the next topic is 17.3, that is exchange of gases. Alveoli are the primary sites of the exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure/concentration gradient. Solubility of the gases as well as the thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented by PO2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in the atmospheric air and the two sites of diffusion are given in table 17.1 and 17.3. The data given in the table clearly indicates a concentration gradient of for oxygen from alveoli to blood and blood to tissue similarity. Table 17.1, partial pressure that is given in mmHg, oxygen and carbon dioxide at different parts involved in diffusion in comparison to those in atmosphere. Respiratory gases, that is O2 and CO2, in pressure, atmospheric air, O2 is 159 and CO2 is 0. 0.3. In alveoli, O2 is 104 and CO2 is 40. In blood that is deoxygenated, O2 is 40 and CO2 is 45. And in also blood that is oxygenated, O2 is 95 and CO2 is 40. And in tissue, O2 is 40 and in O2 tissue, in tissue O2 is 40 and CO2 is 45. And in figure 17.3, diagrammatic representation of exchange of gases at the alveolus and the body tissue with blood and transport of oxygen and carbon dioxide is given. A gradient is present for the CO2 in the opposite direction, that is from tissue to blood and the blood to alveoli. As per solubility of CO2 is 20 to 25 times higher than that of O2. The amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of O2 from alveoli to tissues and that of CO2 from the tissue to alveoli. And next topic is transport of gases. Blood is the medium for transport from O2 and CO2. About 97% of O2 is transported by RBC in the blood. Remaining 3% of O2 is carried out in the dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, where 70% of it carried by bicarbonate as and about 7% of CO2 is carried in a dissolved state through plasma. And the next part is 17.4.1, that is transport of oxygen. Hemoglobin is a red colored iron containing pigment present in the RBCs. O2 can bind with hemoglobin in the reversible manner to form oxyhemoglobin. Each hemoglobin molecules can carry a maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to the partial pressure of O2. Partially pressure of CO2, hydrogen ion concentrated, and temperature are the other factor which can interfere with this binding. A sigmoid curve is obtained when the percentage saturation of hemoglobin with O2 is plotted against the PO2. This curve is called the oxygen dissociation curve, that is given in figure 17.5, and is highly useful in studying the effect of the factors like PCO2, that is H plus concentration etc. on binding O2 with hemoglobin in alveoli where there is a high PO2, low CO2, lesser H plus concentration and lower temperature. The factors are all favorable for the formation of oxyhemoglobin, whereas in the tissue where low PO2, high PCO2, high H plus concentration and higher temperature exist, the conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets dissociated at the tissue Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissues under a normal psychological condition. Next topic is 17.4.2, that is transport of carbon dioxide. CO2 is carried by hemoglobin as carboaminohemoglobin about 20 to 25%. This binding is related to the partial pressure of CO2. Partial pressure of O2 is a major factor which could not affect this binding. When partial pressure of CO2 is high and partial pressure of O2 is low, as in the tissues, more binding of carbon dioxide occurs, whereas the partial pressure of CO2 is low and partial pressure of O2 is high, as in the alveoli. Dissociation of CO2 from the carbaminohemoglobin takes place. That is, CO2 which is bound to hemoglobin from the tissue is delivered to at the alveoli. RBC contains a very high concentration of the enzyme carbonic anhydrase, and minute quantities of the same is present in the plasma too. This enzyme facilitates the following reaction in both directions. That reaction is given in this chart. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBCs and plasma and forms HCO3 minus and H plus. At the alveolar site where as partial pressure of CO2 is low, the reaction proceeds in the opposite direction, leading to the formation of CO2 and H2O. Thus, CO2 trapped as a bicarbonate at the tissue level and transported to the alveoli is released out as a CO2, that is given in figure 17.4. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. And the next topic is regulation of respiration, that is 17.5. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissue. This is done by a neural system, a specialized center present in the medulla region of the brain. Respiratory rhythm center is primarily responsible for the regulation. Another center present in the pons region of the brain called pneumotocentric center can moderate the function of the respiratory rhythm center. Neural signal from this center can reduce the duration of the inspiration and thereby alter the respiratory rate. A chemosynthetic area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substances can eliminated. Receptors associated with aortic arc and carotid artery also can recognize changes in CO2 and H plus concentration and send necessary signal to the rhythm center for remedial action. The role of oxygen in the regulation respiratory rhythm is quite insignificant. And the last one topic is disorder of respiratory system. First one is asthma is a difficulty in breathing causing wheezing due to the inflammation of bronchi and bronchitis. Emphysema is a chronic disorder in which alveolar walls are damaged due to which respiratory surface is decreased. One of the major causes of this is a cigarette smoking. And the last one disorder is occupational respiratory disorder. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis, that is proliferation of fibrous tissue, and this causing serious lungs damage. Workers in such industries should wear protective masks. That's all for today. Thank you for watching this video. Please like, share and subscribe my channel.